I'd like to thank the organizers again. This has been a very unique and pleasurable meeting. I am Jennifer Stone, as was said, and I'd like to talk about a couple cancer immunotherapeutic strategies today and how they compare to one another. Specifically, chimeric antigen receptors and bispecific T-cell engagers. So we can look at cancer as sort of a failure of the adaptive immune response to surveil properly for mutated cells. So in that sense, the immune system malfunction can lead to cancer. We know that in the adaptive immune system, T cells can frequently respond potently to tumors, but similarly frequently they don't. And that's because the normal T cell receptor peptide MHC interaction is susceptible to a lot of different tolerance mechanisms. There's central tolerance because a lot of these tumors' epitopes are very similar to self. There's also immunosuppression effects in a tumor and possibly dysfunctional antigen processing and presentation, causing a lot of these MHC peptide complexes to be downregulated or not present the proper peptides to allow T cells to recognize them. However, it would be very desirable to have these T cells actually respond. So how can we retarget T cells to effectively fight tumors? And I'm going to talk about two strategies today. The first is a chimeric antigen receptor, which consists of a targeting moiety or binding moiety, which is fused to an intracellular signaling domain, all in one chain. And this can recognize a tumor-associated antigen, or TAA, which could be any protein on the surface of a tumor cell, including a peptide MHC complex, as you might have heard earlier from Dave Aggen. The other strategy I'd like to talk about is called a bispecific T-cell engager, or a BiTE. And this is a soluble bivalent T-cell engager that binds to an invariant portion of the T-cell receptor complex and cross-links it to a tumor-associated antigen. So I'll go a little more in depth into these two strategies. The first strategy, CARs, or chimeric antigen receptors, as I said, consists of a binding moiety, which is frequently a single-chain FV, or the binding portion of an antibody, and is fused to these intracellular signaling domains, which can consist of part of the T-cell receptor complex and lots of co-stimulatory signals. And the nice thing about this strategy is we have evidence that it works. In recent clinical trial success, Carl June and his colleagues were able to treat and cure a small number of refractory human leukemias with long-lived anti-CD19 CAR transduced cells. And CD19 is a differentiation antigen present on B cells, both cancerous and regular healthy ones. The second strategy I'd like to talk about is the BiTE, and this is just a little scheme of the BiTE. This would be a single-chain FV against a tumor-associated antigen, which is covalently linked to a single-chain FV against the T-cell receptor complex. And the nice thing about this is it also works. There was recent clinical success for a BiTE called blinatumomab, and this was marketed by Micromet, which was recently bought by Amgen. And in a very small trial as well, 100% objective responses were seen at 100 or 1,000-fold lower mass doses than rituximab, which is sort of the gold standard antibody treatment for B-cell leukemias and lymphomas. And 16 of 21 of their patients became totally negative for minimal residual disease. And that's using this as a single agent. So I'm going to be comparing the sensitivity of these two strategies against a single tumor epitope. And I'll talk a little bit about this epitope and where it comes from. So what we know is tumors present immunogenic antigens. And there have been lots of conserved tumor-associated antigens identified in a variety of different categories. I'll just point out CD19 and CD20 are differentiation antigens that are present on B cells for B-cell leukemias and lymphomas. Overexpressed antigens here are antigens that are normally expressed in healthy tissues but are dramatically upregulated in some tumors. And specifically HER2 I'd like to mention. But if we could get our ideal tumor epitope, what it would be is an epitope that's commonly expressed on a lot of different cancers and yet is exclusively found on tumors and not anywhere in healthy tissue, embryonic, cancer testes, anywhere else. And if we had such an epitope, we would want to use a very sensitive, specific strategy to redirect T cell responses to it. So we have a candidate epitope for this ideal scenario. 237 is an antibody that we're using as our targeting molecule. It recognizes a glycopeptide, which is part of a larger transmembrane glycoprotein. And this is just the crystal structure of the single chain Fv binding to the peptide and the sugar. And the affinity for this glycopeptide is 140 nanomolar. So what we're recognizing is actually an unusual glycosylation, a TN-like antigen, in the context of this OTS8 protein. And OTS8 is a protein that's upregulated on many tumor types, and it's thought to correlate with invasiveness. So this aberrant glycosylation results from a mutant cosmic chaperone. So it's got to have both the OTS8 protein and the cosmic chaperone mutation in order for this epitope to be revealed. This epitope was first found in this murine fibrosarcoma, AG104A. And if wild-type cosmic chaperone is transduced into that cell line, the resulting cell line, A cosmic, is 237 negative. So you can see nice bright staining here and basically nothing here. So what we were able to benefit from is a panel of cells with different levels of 237 epitope. So you can see here I've got 6 cell lines characterized for their 237 epitope staining. AG104A is the cell line I already mentioned with the cosmic rescue mutant. Neuro2A is a cosmic mutant cell line that does not express OTS8, but if you transduce OTS8 in, it becomes very positive for this epitope. And JERCAD is a human T cell transformed line that is also cosmic mutated, and when we transduce OTS8 in, we get a higher level of 237 staining. If you look at this plot here with a titration of 237 staining, you can see that most of these OTS8 cosmic mutant cell lines titrate out at about the same level, but JERCAD, this line here, which is mutated for cosmic and does not express OTS8, seems to have a cross-reactive epitope of a different affinity. So this is another important consideration, that you may have cross-reactive epitopes in the body. So we were able to quantitate these levels by comparison to calibrated beads. And we've got, tens of thousands per cell for Neuro2A and AG104A, just under 10,000 for this JERCAT with OTS8, and this cross-reactive epitope quantitates out to just under 1,000. So now I'll go through the 2 strategies and how we constructed them. First, we have the chimeric antigen receptor against the 237 epitope. And this was actually constructed by Dave Aggen, who spoke earlier today. And we've got the 237 single chain Fv linked to intracellular cytokine, or intracellular signaling domains. And this was cloned into a retroviral vector and transduced into purified ex vivo primary cells. So the cells are conditioned and transduced in vitro and could then be returned into a patient. And we get very high transduction efficiencies in both CD4 and CD8 cells. And by testing, we were able to show that specific responses were seen to 237 positive cells, but not to 237 negative cells. So for comparison, we also constructed the bispecific T cell engager to this very same epitope. And here we have 2C11, which is an anti-CD3, so anti-T cell receptor complex antibody. The single chain Fv of that linked to the single chain Fv of 237. And this protein was purified from mammalian cells, and we get nice pure protein and high yield. If we test just the anti-CD3 portion by coating on a plate, we see that the anti-CD3 single chain Fv stimulates very similarly to the full antibody. And if we then put it in with T cells with an irrelevant T cell receptor, we can take those T cells and make them lyse 237 positive cells specifically. So this strategy also seems to be working. Finally, I'd like to compare directly side by side these 2 strategies. So here I have a slide of cytokine release induced by 237 targeting strategies. And here we're looking at just AG104A and A cosmic. so the wild type and the cosmic rescue mutant. On the left, I have T cells targeted by 237 CAR, and on the right, 237 BITE. And then for each, we have CD8 and CD4 tested. So what you can see is we have specific responses all the way across this panel, but we have decreasing sensitivity as we go from CAR to BITE and from CD8 to CD4. If we expand this to the full panel of cells, we see the same trend across the rows, but we also see a trend of decreasing sensitivity as we go from high antigen levels per cell to lower antigen levels per cell. However, if we look at a different response, and instead of cytokine release, we look at target lysis, I'd like to point out this Jurkat cell line, which had the secondary cross-reactive epitope. And now, even though this is OTS8 negative, we get very efficient lysis by especially 237 CAR redirected T cells, and even a little bit by BITE T cells. So this epitope does cause responses from both targeting strategies. So given all of this data, we were able to construct a sensitivity scale for targeting this epitope via CAR versus BITE. And this is just a summary of what we've seen before. Cytokine release is less sensitive than target lysis. CD8 is more sensitive than CD4. And CAR is more sensitive than BITE. And each bar is stretching from high antigen levels down to the minimum antigen level per cell we expect to see the response. If we look at other tumor-associated epitopes on this same sensitivity scale, we can see CD19, which is the epitope that is being targeted by both the clinical CAR and BITE strategies I mentioned earlier. And you can see it would be very efficient for target lysis, but you might not get quite the cytokine release from CD4 or CD8 BITEs that you would from the CARs. I'd also like to mention HER2. HER2 is an upregulation antigen for breast cancer. And here we have the levels on normal breast, and you can get up to a 40-fold increase of level on about 15 to 30% of tumors. This normal level is already within the sensitivity range of both CAR and BITE. So it's very important to know this sort of level before you approach a strategy like this. And in a very unfortunate incident, HER2-directed CAR T cells were inserted into a patient, resulting in very rapid lethal effects due to expression of HER2 on the lung. So in conclusion, we were able to have T cell targeting to the 237 epitope, which is specific via CAR or BITE. Stimulation of transduced T cells with 237 CAR is more sensitive than stimulation mediated by the BITE, and that was across the board. And then CD4 T cells were active in the same assays as CD8 T cells, although they were less sensitive also across the board. And the choice of CAR or BITE will have to depend on the antigen density, specificity of the epitope expression, and of course other factors such as ease of delivery and manufacture, etc. I'd like to acknowledge the members of the Kranz lab, especially Dave Aggen, who constructed the 237 CAR, our collaborator Andreas Schedinger, who was the characterizer of the 237 epitope and made several of these 237 target cell lines, our ongoing collaboration with the Schreiber lab at the University of Chicago, helping us move in vivo with some of these targeting strategies, and our funding from the Cancer Research Institute and the NIH. Thank you very much, and I'll take any questions.
I have a quick question.
Okay.
I don't really understand the difference between CD4 and CD8 population and how they were listed different infections. Like the less sensitivity in the CD4 population. Do you know what mechanism causes that?
We're not really sure why they're less sensitive in this case, since these are not co-receptor dependent reactions. But we did see this in all of the cases that we tested, that both target lysis and cytokine secretion were less potent from the CD4 T cells. And that could just be based on the way that they function normally.
All right, I think let's thank our speaker once more, and then we'll bring up the next student.